Nathan and Joyce and the rest of the team for setting up this event. This is really wonderful. And I also want to thank Thore for setting up some of the themes I'm going to discuss so nicely. So what I want to do today is just introduce some of the work that we're doing at Valence Labs towards this grand vision of autonomous scientific discovery. The research programs that we're working on are really focused on asking the question of, can we teach these computational systems to run experiments, analyze data, generate hypotheses, develop predictive theories in a way that not only matches human capacity, but ultimately exceeds it. And so just really quickly about me, as Nathan mentioned, I was co-founder and CEO of a company called Valence Discovery. We were doing ML and physics-enabled molecular design. Just over a year ago, we were acquired by a company called Recursion. For those who don't know Recursion, it's probably the leader in tech-enabled drug discovery and development, publicly traded company, 5 medicines in clinical development, very well resourced to pursue this mission of industrializing drug discovery. And we came together to form an organization called Valence Labs, which is an independent AI research group within Recursion. And what I want to do today is just share a little bit about our story at Valence Discovery as a startup. I suspect there are several founders or future founders in this audience. Talk about some of the stuff we learned, some of the stuff we did right, some of the stuff we did wrong. Transition into how that led us to the team at Recursion, and then close with some of the work that we're doing at Valence Labs. So Valence Discovery was a team out of the Montreal Institute for Learning Algorithms. Very fortunate to work closely with folks like Yoshua Bengio as close advisors. We were all grad students at the time that we founded this company. We were working with Pfizer and Merck on problems in peptide design and therapeutic design. And we, through that process and some serendipity, were able to put those grad programs on hold and start a company focused on therapeutics discovery. I think we've heard from the speakers this morning, I hope everyone's getting a sense of how complex this process of discovering medicines actually is. It really should not work when you take a step back and think about all the different steps involved in bringing a medicine to patients. And me personally, I have history of neurodegenerative disease on one side of the family, history of cardiovascular disease on the other side of the family. Like, what better way to apply our technical skills to problems that matter than starting a company focused on this space? And our philosophy at the time, which is, I think, now kind of a consensus view, but at the time fairly controversial, was that drug discovery is inherently a low data problem. We had so much interest in this space when we were starting the company in 2018. We had so many people entering this space, trying to apply methods from other areas of machine learning to this problem of drug discovery, talking about how much data there was available in this space publicly. But the reality is there really wasn't that much data, and there definitely wasn't a lot of high-quality data. And there are various flavors of this low data problem. I think Thore introduced this really well with these different levels of complexity in this process of bringing a medicine from kind of an early hypothesis to a patient. This is one of these flavors. We generally have in drug discovery an inverse relationship between the quantity and quality of data, quantity of data and the translational relevance of that data. When you're making a medicine, you can't just have a molecule and bring it directly into a human. You need to go through all these intermediary steps before you get there. You start with these very simplified models of disease biology, and over time you work towards more complex models of disease biology. You eventually move into animal systems, and you move into the ultimate goal, human systems. And what we find is that we have loads of data available for these very, very early parts of the drug discovery process, these very, very low fidelity systems that give you only a kind of slice of the total complexity of the systems that you're trying to modulate. And we have far fewer data at the other end of the scale. We're looking at these more complex systems. And there's other flavors of this. Generally, when you're running a drug discovery program, you end up with these very sparse data matrices. You can kind of just take a step back and think of molecular design as a multi-parameter optimization problem. You can think of it as kind of a whack-a-mole problem. If you picture your drug as this multi-faced Rubik's Cube, typically what happens in a drug discovery program is you solve one face of the cube, then you move on to the next face, and solving that second face comes at the expense of the first face. And you work in these kind of iterative loops of trying to solve all the faces of this cube, optimizing for all these different properties that you need to solve for in order to bring a molecule into human clinical testing. We also find that for many of these molecules, we end up with far, far, far more that don't work than those that do. So we end up with these hugely imbalanced data sets that are rather difficult to work with from a machine learning perspective. And so at Valence Discovery, the approach we took was one of algorithmic innovation. We believed that if we worked hard enough, we could kind of make our way through some of these very complex technical issues. We were doing a lot of work in areas like few-shot learning, meta-learning, representation learning, active learning. And we published a lot of that work very openly. And in doing so, we were able to build up, I think, a pretty strong research brand for ourselves. And as we published more and more of this work, we started to get more inbound from potential recruits. We started to get more inbound from potential investors. Started to get more inbound from potential pharma partners that wanted to use these tools in their real-world drug discovery programs. And so we did end up signing a number of deals with large biopharmaceutical companies. And we were able to take these methods and apply them in real life. This was really cool. So what's shown here is a bunch of these different parameters that you need your molecule to satisfy. You have things like activity, toxicity, stability, metabolism, permeability. These are all these different properties that your molecule needs to, or criteria that your molecule needs to meet in order to be considered as a drug. And what we did with this pharma company is we came into one of their drug discovery programs. We took the best molecule that their chemists had been able to produce in the 2 years or so that they've been working on this program, as delineated in the orange here. So you see that it satisfies some of these parameters, but not all of them. We're able to take all that data and use it to design new molecules. We have those molecules synthesized, tested in the lab, and we find that they're able to satisfy a broader set of these parameters. And we did all sorts of work like this with biotech companies, large pharma companies. We had an internal drug discovery pipeline in oncology that we were pushing forward. And it was really cool to take these research methods and apply them in real-world drug discovery programs. Along the way, I suspect I'm not the only one here who's gotten a message like this, but along the way, Nathan reached out, wanted to learn more about our work, and over a period of, 12 to 18 months, had eventually led to an investment in our seed financing. So thank you, Nathan. But the backdrop against which we were operating was constantly evolving. What was happening in 2018 was very, very different than what was happening in 2021, 2022. And there were these, every day we were seeing a new kind of scaling laws related paper. The general idea here is that bigger is better. More data, more compute, leading to better performance. And as CEO of this company, every day I was waking up and I was wondering, is this the day that OpenAI drops a model that makes a whole generation of work that our team has been doing obsolete? And so as a team, we started thinking about how can we adapt to this new landscape that we find ourselves operating in. And so we started thinking about data sets that we would need to generate for ourselves to remain competitive, to differentiate from our peers that were training on the same corpus of undifferentiated public data. We started thinking about lab space. At the time, we were working with these external contract research organizations, so these external labs that are running our experiments for us, working on these 2 to 4 week turnaround times. You can imagine that if you have to wait 2 to 4 weeks for every piece of data to come back to you, your cycle time is going to be very slow. So we started thinking about what kind of lab space would we have to build for ourselves. We started thinking about compute. We were emailing everyone, trying to beg, borrow, and steal our way into more compute to scale up our models. Started thinking about how much money we're going to have to raise to build the infrastructure we need to succeed at the level we want to succeed at. And so we were preparing to go out and raise $50, $60, $70 million to resource our team appropriately to pursue this mission. And it was around that time that we started talking to our friends at Recursion. So Recursion is a company that's been around for over 10 years. And the basic thesis of the company was that we need to generate our own data sets in a highly controlled, fit-for-purpose manner to enable or to realize the real potential of machine learning and drug discovery. So Recursion has spent over a decade generating huge amounts of highly controlled data sets that they're able to train on to drive downstream drug discovery programs. I think Recursion is at something like 25 petabytes of internally generated drug discovery data across biology, across chemistry, across early discovery through clinical translation, running up to 2.2 million experiments per week. I think Recursion generates every 15 minutes the equivalent of an average PhD student's worth of data. So they had that piece under control. Recursion also has highly sophisticated automated lab infrastructure to generate additional data sets in perpetuity, but also to test the computational predictions that we're making in our labs with very short turnaround times. And finally, Recursion, thanks to our partners at NVIDIA, there's a huge amount of compute available. We've recently brought online our newest cluster with close to 1,000 modern GPUs available to our team, top 35 supercomputer, and by far the largest cluster in the biopharma industry available for our teams to train on. And so we decided to come together and form an organization called Valence Labs. And Valence Labs is an independent AI research group within Recursion. We're somewhere between 40 to 50 people split between Montreal and London, growing fast. As Nathan mentioned, we just opened our London site officially yesterday and expect to fill that office with another 10 to 20 people quite quickly. And we have this really, I think, ambitious, inspiring, audacious mission of industrializing scientific discovery. And our vision for the future, if we're successful in industrializing scientific discovery, is that we'll be able to take this process of therapeutics discovery, of scientific discovery, and do it at scales that were previously unimaginable. And so the way we frame this is that we want to develop highly autonomous systems capable of Nobel-worthy insights. And if we can do that, if we can run the scientific process computationally with close integration with wet lab and human scientists, we can usher in a new era of therapeutic discovery. And so we break our research program, our long-term research program, into three distinct parts. We have foundation models of biology and chemistry. So how can we capture the complex worlds of biology and chemistry computationally? Inference engines and active learners. How can we most efficiently use the wet lab infrastructure we have available to us to validate hypotheses, to run experiments? And orchestration and reasoning systems. How can we take all of these pieces and orchestrate them together in a way that allows us to run these workflows far more efficiently? And so I just want to quickly walk through each of these areas and share some of the work that we're doing at Valence Labs. So this is work from our team in learning foundational representations of biology. So the basic question we're trying to answer here is, can we capture the complexity of biology in a computational representation? The task here is to reconstruct these cellular images as accurately as possible. And the idea is that the better job we can do at reconstructing these images, the more useful these representations are in downstream tasks in drug discovery at Recursion. And this is a little bit blurry, but when you blow these images up, it's actually truly remarkable how close these reconstructions come to the original images. And as we were working on this project, what we found was that as we threw more data at these problems, we threw more compute at these problems, performance improved. And performance is continuing to improve in work that we're doing internally as we continue to scale these projects up further and further. We've taken similar approaches at learning computational representations of the chemical world. So this problem of how do you best represent a molecule computationally is a very active area of research. Folks like Michael have done a lot of work in this space. And the idea is that we want to capture the very complex physical interactions between molecules and their environment in a computational form. And again, what we find is as we throw more data at these problems and more compute at these problems, performance improves and continues to improve as we scale these models further. And what's really cool, this is very recent work from our team just submitted at NeurIPS, but what's really cool is we can take these representations of biology, take these representations of chemistry, and build basically the equivalent of DALL-E for drug discovery. If you think about the architecture of early DALL-E models, about joint image and text embeddings, we've basically taken the same principles and applied them here, but with embeddings of biology and chemistry. And what this allows us to do is for any given molecule, we can get an understanding of the biological kind of effect that that molecule will have in the system of interest. And for any given perturbation, biological perturbation, we can get a molecule that would yield that result. And this is really, really cool. This allows us to kind of move seamlessly between the worlds of biology and chemistry in a way that powers our drug discovery processes at Recursion. So once we've built these representations of biology and chemistry, we want to query them in some way to run experiments in the real world. And the basic principles that we use at Recursion are to use active learning as a tool to run these experiments more efficiently. Even with all the experimental capacity we have, we cannot run every single experiment. We need to be smart about which experiments we choose to run in the lab. And so the general philosophy that we take is to predict where we can extrapolate, but experiment when we can't. And I have just a toy example here to illustrate what we mean. So let's say you have a cell, and in the cell there are a number of genes, and we want to study the octopus gene. And we might choose to upregulate or knock out the octopus gene, and the result is that we get this octopus cell. If we choose to knock out the Mr. Monopoly gene, we might get this cell. And if we choose to knock out the unicorn gene, we might get this cell. And sometimes we want to look at the effect of playing with more than one gene at the same time. So we might want to look at what happens when we knock out the octopus gene and the Mr. Monopoly gene. What is the result that we get? We might want to knock out the Mr. Monopoly gene and the unicorn gene and see the result that we get. But what about when we knock out the octopus gene and the unicorn gene? Is it going to be an octocorn? Is it going to be a unipus? We don't know. And the idea is that we want to run physical experiments, or we want to prioritize our physical experiments in these areas of high uncertainty. And so this is work from Jason Hartford and his team, but the fundamental question we're trying to ask here is, can we recover the majority of the information from only a subset of the experiments? And what's shown on this slide above, these yellow squares represent areas of highest uncertainty. What we find is an active learning approach does far better than this random search approach in uncovering these areas of high uncertainty, allowing us to redirect our platform resources far more efficiently. And finally, when we have all these pieces, we want to be able to orchestrate them in some way. If we want to be developing or working towards a future in which agents are running all of these pipelines independent of human input, we need some degree of orchestration. And we have, currently, the approach we're taking here is to use LLMs as orchestrators of these complex workflows. LLMs today are not yet at a point where you can ask them questions, fundamental questions about biology and chemistry and get a good result. You can't go to ChatGPT and say, design me a drug for cancer indication A. You won't get what you expect. But you can use these LLMs as orchestration systems. You can use them kind of run through complex workflows with far less human input that you'd otherwise require. And so at Recursion, we have all these different tools in our drug discovery platform. And typically, these tools require very specialist human input to run correctly. And for different problems in drug discovery, we might use different set of tools. So for example, we may use this set of tools if we're trying to design a new medicine for a novel disease area. We might use a different set of tools if we're trying to repurpose an existing medicine for a known disease. And one of the projects that we worked on within Valence Labs was this idea that we could use LLMs to orchestrate all of these complex workflows in a manner that requires very little human input. And the idea here is that we can move from a world of scarcity, where you previously required specialist training in machine learning to run these workflows, to one of abundance, where any biological or chemical scientist without any training in computer science can run very complex computational workflows. And the takeaway here is that at some point in the not too distant future, we believe that we're going to have these relatively autonomous systems that are able to generate hypotheses, design experiments, learn from the results, automatically repeat these processes 24/7, 365 in a way that can create far greater productivity in drug discovery than we'd otherwise be able to accomplish. And just really quick plug, if this sounds interesting, we have a very active research community that we manage and run. We have weekly reading groups in geometric deep learning, molecular modeling, causality. We have a huge number of open source tools that our team has released, focused mainly on cheminformatics and molecular modeling. And you can follow all of this at portal.valencelabs.com. And we also have a number of open positions across Recursion and Valence Labs at our new London site in King's Cross. Thank you.